Thank you for the introduction. Good afternoon, I'm David Rodriguez, and I will be discussing the application of Fourier Transform Infrared spectroscopy technique in our hospital. As you know, FTIR spectroscopy is an emerging technique in clinical microbiology that is gaining popularity, particularly in bacterial typing. Furthermore, we are currently in the era of artificial intelligence, as you see every day in the media, and we can use this tool for multiple purposes. For example, not only you can use AI for image creation, but also for the analysis of FTIR spectroscopy data, as I will show you during this presentation. I have divided the content of the presentation in several parts. To begin with, I will provide a brief overview of the principles of FTIR spectroscopy, followed by an explanation of the sample preparation process and the analysis of the spectra. Then I will show you several examples of studies conducted at our hospital, specifically focusing on the characterization of outbreaks caused by 3 different microorganisms and on the classification of strains. Finally, I will conclude the presentation by summarizing the key findings and implications. To introduce this technique, it's essential to understand that it is based on the interaction between infrared light and matter. When we irradiate a sample, it absorbs the infrared light, and the frequencies of the absorbed light vary depending on the compounds present. How does it vary? Because the molecular bonds of the compound absorb the infrared light at different frequencies, and it generates vibrations. The Fourier transform is the mathematical process that decomposes a signal into all its frequencies, enabling a better visualization and analysis of the data. So, how can we apply this technology to bacteria? Well, all bacteria are formed of various components such as carbohydrates, lipids, proteins, and these compounds can absorb the infrared light. Depending on the specific structure and composition of these molecules, the frequencies at which they absorb the light are different, and then the FTIR spectrum obtained is different and highly specific. This characteristic enables us to differentiate between closely related microorganisms, and therefore we can identify specific bacterial strains. This process involves simple and rapid sample preparation and is universally applicable on bacteria and also yeasts. Now we will take a look closer into the spectrum. The vertical axis represents the absorbance, while the horizontal axis represents the wave number. This wave number is the measure of the number of waves per unit distance, typically expressed in centimeters, and it is inversely related to the wavelength of the light. This parameter is directly proportional to the energy, meaning that higher energy corresponds to higher wave number. Along the spectrum, there are different regions that are related to different compounds found in the cell. For instance, the ER biotyper defines a default region between 800 and 1100 centimeters that corresponds to carbohydrates, which is commonly used for bacterial typing. There are also other regions available for analysis, such as the lipids region, which includes 2 sections of the spectrum, the proteins and carbohydrates region, or the humidity region, which is also represented by proteins. In addition, it's worth mentioning that users have the flexibility to select or create other customized sections of the spectrum based on their specific needs or interests. As we have seen before, this is the typical FTIR spectrum. However, when we are analyzing different strains and we aim to find differences between them, working with the raw spectrum becomes challenging due to the overlapping data, and it becomes very difficult to distinguish these minimal differences. In order to solve this problem, the system employs the second derivative of the spectrum. Up here you can see the second derivative of the entire spectrum, but now if we take a look to a specific region, such as carbohydrates, we are able to see differences more clearly in the spectra of the samples. The second derivative analysis considers the slope and the curvature of the spectrum and provides us a new one with an enhanced resolution. Now, with this refined information, we can work with the data. Now, let's move on to the sample preparation process. If you have already worked with this technique, you are likely aware of its simplicity. Firstly, we need to resuspend the bacteria in ethanol and mix it thoroughly using a vortex mixer along with the metal cylinders of the tube to achieve a homogenized solution. There are two critical factors to consider during these steps. First, the amount of bacteria is crucial. It is necessary to obtain an overload loop of 1 microliter. And second factor is the homogenization. Achieving a complete homogenization is vital in order to avoid failures of the system. After the ethanol step, we add water, vortex the mixture again, and finally we deposit 15 microliters of the sample onto the plate. There are some microorganisms or certain strains that are difficult to homogenize, and you may require an alternative protocol. There are two of them. One protocol involves a direct smear of the bacteria on the sample plate, followed by inactivation using ultraviolet light. And a second alternative protocol directly homogenizes the bacteria in water. You can also inactivate the bacteria by ultraviolet light in this procedure. Once you have homogenized the samples, you can proceed to deposit them on the plate. Typically, we deposit the samples in triplicate, which is the minimum number of replicates indicated by the system. Then, the maximum number of samples that can be analyzed on a single plate is 30 samples. For each run, it's necessary to include 2 control samples in duplicate and 1 blank spot. That results in a total of 95 spots used. After deposition, you can either dry the plate in an incubator or at room temperature. In any case, it's important to ensure that the samples are completely dry before proceeding with the analysis. For a correct analysis, each spot must pass the quality test of the system. In the event that a parameter fails, you can repeat the analysis for that strain. In our experience, the absorption parameter is often the cause of most failures, and it is associated with the amount of biomass taken from the initial culture. If you apply too much biomass, it will absorb a lot of light and the value will be above the maximum. And if you don't take enough biomass, the absorption will be too low. Now it's time to analyze your data. You have two options for analysis: unsupervised methods or supervised methods. For unsupervised methods, there is the principal component analysis and hierarchical component analysis, which are two methods widely known and used. On the other hand, for supervised methods, there is available the linear discriminant analysis. The main difference between these approaches lies in their underlying principles. With the unsupervised methods, the system analyzes each spectrum individually and creates a representation that reflects the distances between them. These algorithms don't consider the labels or the categories of the samples. In contrast, the supervised methods rely on the labels or categories you have selected for your samples. This means that the algorithm will try to search specific differences among the labels in order to create a representation that effectively separates the samples in distinct clusters according to the labels. After an initial screening with the previous methods, if you observe a satisfactory separation of clusters, you can proceed to create a classifier based on AI. The purpose of a classifier is to automatically identify a new sample according to the categories defined within this classifier. It can be made with 3 different supervised algorithms: artificial neural network, linear support vector machine, or radial basis function SVM. Don't be afraid of the names of the algorithms. The important point is to understand that each algorithm will analyze your data in a different way, and one of them may be suitable for the purpose of your study. The creation of a classifier involves 2 main steps. In the first step, you need to train the classifier using a group of samples that encompass all the variability that we aim to separate, this set is referred to as the training group. And through this process, we will obtain a confidence value that we call internal validation. Then, to evaluate the performance of this classifier and be sure that it works well, we select a new group of samples and we use the classifier to identify them and see if they are correctly classified. This external validation indicates the number of samples that have been correctly assigned to their respective categories. Now, in the upcoming slides of the presentation, I will show you several examples where we have applied FTIR spectroscopy. The first one is an outbreak of Pseudomonas aeruginosa that we analyzed retrospectively. This particular outbreak involved 14 patients, and in the analysis we include 15 outbreak isolates and 5 unrelated isolates. Prior to our study, this outbreak was characterized using pulse field electrophoresis and whole genome sequencing. With the latter method, we detected 3 subgroups inside the outbreak according to single nucleotide polymorphisms. And here is the dendrogram obtained using the default region of the system, which corresponds to carbohydrates. This outbreak was caused by a Pseudomonas strain belonging to ST175, and we can observe that the majority of this ST was in the same cluster. However, one outbreak strain was classified outside this cluster together with other STs. If you remember from previous slides, we have the ability to perform the analysis in different regions of the spectrum. So in this case, if we take a look to the lipids region, we obtained a different dendrogram where all outbreak isolates were correctly grouped in the same cluster. It's worth noting that 2 isolates of this study belonged to the same patient, and they were grouped together with a very small distance between them, indicating their close relation. We can also visualize a similar representation using a PCA, also in the same lipids region. In this case as well, the clusters were also clearly differentiated. If we try to differentiate the subgroups observed by whole genome sequencing, they were not clearly distinguishable by FTIR spectroscopy. But in any case, it was still useful for accurate classification of all outbreak-related isolates. So from this study, we conclude that FTIR spectroscopy achieved the same discriminatory level than PFGE, and although the discriminatory level was not as high as that of whole genome sequencing, it identified successfully the outbreak isolates. It is important to note that during the initial studies of a microorganism, it is advisable to explore various analyses, visualizations, and results obtained with different regions of the spectrum. This allows us to determine the most suitable combination for future studies involving that particular microorganism. The next outbreak I will present was caused by Elisabethingia miricola, which is an uncommon microorganism. Usually we detect a few number of isolates of this species each year, and during this 2 years period we detected up to 23 patients infected with Elisabethingia. In this case, the FTIR analysis was conducted before the molecular characterization and during the last months of the outbreak. So some isolates were analyzed in real time at the moment of the isolation in the laboratory. In addition to the clinical isolates, we obtained 3 more environmental isolates from the sinks in several hospital rooms. Initially, we performed PCA using the default region, and we observed these clusters. You don't need to be an expert to notice that something is happened here. There are a lot of isolates clustering very closely, while 3 isolates are located outside this cluster. So we can suspect that all this cluster could potentially represent the outbreak strains. Then we perform the dendrogram analysis. The first important point is that one isolate was not an Elisabethingia miricola species, it was an Anophelis identified by Malditov. So this patient was not related to the outbreak. The other 2 isolates were significantly distant from the main cluster, so it could indicate that these two isolates were also not related to the outbreak. To confirm these findings, we perform whole genome sequencing, and this is the representation of core genome MLST. First, we observed that the outer isolates were almost the same, isolates number 4 and 18, and I will discuss later the isolate number 5. This small cluster inside the outbreak corresponded to this group of isolates, while the remaining isolates belonged to all this central group. Furthermore, the 3 environmental isolates were also included in this central group, and therefore we could confirm that the environmental and the outbreak causing strain were indeed the same. When comparing FTIR spectroscopy with whole genome sequencing, we observed one discrepancy, which was strain number 5, while FTIR classified it as a part of the outbreak, as sequencing indicated it was unrelated. However, this could be considered a minor error by FTIR, because although this strain was considered as part of the outbreak, the most crucial result is that this technique didn't miss any outbreak isolate, which is essential for outbreak monitoring and control. So, as a conclusion, FTIR spectroscopy showed a high correlation with sequencing, and the environmental isolates, which were analyzed in real time during the study, were rapidly linked to the outbreak. And the last outbreak I will present was a little bit more complex. This was caused by Klebsiella oxytocca carrying the VIM carbapenemase resistant mechanism, and it primarily affected children. Also in this case, we performed the FTIR analysis before molecular characterization. For this outbreak analysis, we included all the Klebsiella oxytocca VIM isolated detected during this period of time. It encompassed a total of 21 patients, consisting of 15 children from our hospital, 1 child that came from another hospital, and 5 adults. In addition, during the study with FTIR, it was isolated an environmental strain. To ensure accurate analysis, we also include control strains that were clearly not related with the outbreak. 1 susceptible isolate, 1 carbapenemase resistant isolate with OXA mechanism, and 1 VIM isolate obtained in previous years and was most likely not related with the current outbreak. This is the final dendrogram we obtained after the analysis of all the described isolates. Let's see the placement of each isolate I have previously described. We start with control strains, the susceptible, the OXA, and the old VIM isolate. That's good, they are far from each other. And next, if the outbreak was occurring in children's ward, let's see now the adult isolates. It seems that there are not important relations between them. Moreover, the isolate of the child coming from another hospital was also very far. And moving on, as you may have noticed, there is a main cluster in the center of the dendrogram, which raises suspicions. However, there are still 2 other isolates located outside this cluster, and interestingly, they are both children isolates. So this suggests that these 2 isolates are not related to the outbreak, whereas all the remaining children isolates belong to the same strain, and therefore these patients were involved in the outbreak, including the environmental isolate. The whole genome sequencing confirmed these results, and no discrepancies were found. Now let's see the same representation obtained by PCA. In this visualization, there is a group of isolates here, but it seems that some isolates are quite close together, and it would have been difficult to decide if they were related or not to the outbreak, only with this visualization. In PCA, it's very important to explore all possibilities. For example, the visualization of data in 3 dimensions. This is the image with the same orientation as the 2 dimensions plot. And here you can see close isolates that could be confusing, but when we move the perspective, we can now clearly see a group separated from other isolates, and that blue cluster was the outbreak. So in conclusion, this study, the classification of the outbreak by FTIR spectroscopy matched with the results obtained by sequencing methods. The next example I want to share is about the classification of subspecies using a classifier. In this case, we aim to distinguish between three subspecies of Mycobacterium abscissus. There are some microorganisms that are difficult to work with them, and this is one such case, primarily due to different morphology of the colonies. As I mentioned earlier, achieving a well-homogenized bacterial suspension is crucial during sample preparation, and this is especially difficult with rogue strains of Mycobacterium abscissus. During the acquisition of spectra, we obtained a lot of failures due to a bad homogenization of these colonies, and it was common to repeat most of their analysis. In this example, PCA and dendrogram analysis were not successful in separating the subspecies, but we obtained these clusters using LDA, which is a supervised method. It suggests that the system is able to find differences in the spectra among these three groups. It's important to note that this was a pilot study with a small number of isolates, and it requires further improvement. Now let's imagine that we aim to create a classifier in order to identify automatically new isolates. And for this task, we need the AI option for the creation of effective classifier. If you remember, there are different algorithms available for the creation of a classifier. In this case, we selected the ANN. Before training the classifier, we should decide how many principal components we want to include. Briefly, principal components indicate the amount of data we can use to cover most of the variability of the samples, and it is possible to visualize a graphic with the contributed variability of each component. For instance, selecting 12 to 14 components would be enough in this case, but you can try several options to find the best choice. Now we can train the classifier. Through this process, we will obtain a confusion matrix that reflects the accuracy of this classifier. This value is the internal validation, which indicates how accurate the model performs using the samples used for its creation. The next step would be the validation of this model using a different set of strains to assess if they are correctly classified by the system or not. We have not performed this analysis yet because we have been too busy with outbreak analysis, and because for the creation of a good and robust classifier, you need a large amount of strains and spectra. The ER biotyper includes other pre-existing classifiers that can be used with your strains. And finally, this is the last example made with Klebsiella pneumonia. One of the objectives was to differentiate between some STs, but I think that the most interesting part of the study was the aspect of reproducibility. For this purpose, we analyzed the same group of isolates in two different hospitals, and this involved different personnel and two different IR biotyper instruments. In total, we analyzed the same 76 isolates in both hospitals, and then we combined the results. This led us to ponder the following questions. If I made a visualization of this data, for example, a PCA, they will form clusters according to the STs, or they will form clusters according to the two centers due to the variability arising of personnel and instruments. So here is the PCA we obtained. As you can see, there are some distinct clusters that correspond to a specific ST, particularly those with a higher number of isolates included. Additionally, the spectra acquired from the two hospitals don't overlap, but they are very close each other. For instance, this is the spectra of strain number 20 analyzed in hospital 1, and over here we have the same strain analyzed in hospital 2. And the same happens for other isolates. The next step would be the creation of a classifier, as I have shown you before, and the addition of more isolates of these STs and maybe other difference. If we take a look to the three-dimensional view, here is a video where you can see that if I move the camera around, there are several STs located in the center, but some of them are grouped closely with others of the same category, such as the white cluster and the light blue cluster. So this study showed that the method is reproducible, indicating that the results from different hospitals can be shared in order to perform multicentric studies or to create a larger and more robust classifier. Moreover, it was possible to separate those STs with a higher number of isolates using simple analysis like PCA. So finally, based on all these studies, we conclude that FTIR spectroscopy is a rapid technique with which it's possible to characterize outbreaks, and most importantly, it could be used as a first screening tool before employing more accurate molecular methods that need additional time to generate results. This technique has demonstrated reproducibility and very easy to standardize, offering simplicity and requiring limited hands-on times. The software used for spectroanalysis and data interpretation is user-friendly and is not necessary expert knowledge in machine learning or AI. And finally, further research is needed in different groups of microorganisms, as each species needs their own parameters and their own analysis. That's all from my part. I would like to thank Brooker for inviting me to this webinar. I hope you have found some of these examples interesting and that they could be helpful for your work and your knowledge. Thank you for your attention.